Hello, my name is Lars Bode. I'm a professor of pediatrics at UC San Diego in the United States, and I'm going to talk about the potential of human milk oligosaccharides, short HMOs, what they are and what they do. Before I start, a quick introduction to disclosures, and let's dive right in. I'm starting the presentation with this sentence from the Lancet Breastfeeding Series 2016 that reads that the death of 823,000 children and 20,000 mothers each year could be averted through universal breastfeeding, along with economic savings of $300 billion US. And the question for us is always, why is that? What is so powerful that it can save lives? What is in human milk that drives this? And how does this actually work on a mechanistic level? Let's lift this statement and see what is behind the curtain here. And if you look at the macromolecules in human milk, we see there's protein, fat, lactose, and then there is oligosaccharides. 5 to 15 gram per liter, which often exceeds the total amount of protein in human milk. If we compare that to cow milk, there's about 100 to 1,000-fold less oligosaccharides in cow milk. And since cow milk is the basis for most infant formula, we currently have hardly any human milk oligosaccharides in infant formula. So there's a huge difference between breast milk and infant formula. So what are human milk oligosaccharides? Let's talk about structure for a second, and I'm not going to try to blow your mind here. Oligosaccharides means oligos, a few, and saccharide, sugar. So a few sugars is what oligosaccharides means. When we talk about sugar, we often think about the table sugar that we consume plenty of. And in the infant space, of course, that is not what we want. As a chemist, table sugar looks like this. That's not human milk oligosaccharides. Human milk oligosaccharides are way more complex than that. So here's a structure of a fairly simple human milk oligosaccharide. And if we had to draw this out every time, it would get very tedious and very difficult to understand. So we give every building block, every monosaccharide here, a different structure. And then we can talk about human milk oligosaccharides using these different symbols. And there's really only 5 building blocks to human milk oligosaccharides. There's glucose and galactose, which we know forms lactose. And then in addition, we have N-acetylglucosamine, fuel, and sialic acid. So this is almost like Lego bricks. Depending on how we put those building blocks together, we get either a little car or we get a little plane. And we all know that cars and planes do different things. And that's the same with oligosaccharides. Depending on how we put those building blocks together, we get different structures, and different structures do different things. Structure determines function. Just to give you a few examples here how this looks like, in the left upper corner, I'm going to develop what we call the blueprint of human milk oligosaccharides. And then in the white space around it, I give you a few examples for oligosaccharides. So all human milk oligosaccharides start with lactose at the reducing end. They can be extended by disaccharide units in different linkages and extended even more. And you see this gets fairly complex already. You can get branch structures or long linear molecules. Then these oligosaccharide backbones can be fucosylated either in alpha 1,2, 1,3, or 1,4 position. If we do that on lactose alone, we get 2-fucosyl lactose, or short 2FL, and 3-fucosyl lactose. And we can extend or add fucose to the extended chain and then get different fucosylated oligosaccharides in different linkages. In addition to fucose, we can also add sialic acid. And sialic acid now introduces a negative charge to oligosaccharides. Again, we can add sialic acid simply to lactose to get 3-sial-lactose and 6-sial-lactose, or add it to the growing complex chains in different linkages and then get different oligosaccharides that carry one or more sialic acid, so more negative charges. Overall, we have about 150, 200 different human milk oligosaccharides that have been identified so far. And the key is that different moms make different oligosaccharides in different individual variation. You see this here from the child cohort, different subjects, about 1,200 different women that were analyzed here. And different colors are different oligosaccharides and the different concentrations. And you see how different that is between the different moms. And even if you take all the different oligosaccharides as 100%, even there you see the pattern is very different. So it's almost like a thumbprint where different moms make individualized different oligosaccharides at different concentrations. That's remarkably constant over a short period of time. So here we have samples from the same mom taken daily, and you see really how the oligosaccharides are consistent over a week. And we went even smaller in resolution here, high in and did this every hour. And again, the oligosaccharides are remarkably constant. That is different between two different subjects. So the subject on the top is different from the subject on the bottom. And again, it's different between the two different subjects, but remarkably constant over time in a short period of time. However, they change over the long period of time. So between 2 months and 6 months, for example, there is quite a difference in oligosaccharides within the same. Here's a different representation of human milk oligosaccharides now looking at principal component analysis. So these are human milk oligosaccharide compositions from about 10,000 different women. And every dot represents one milk sample and the oligosaccharide composition in it. The closer those dots are in this three-dimensional space, the more similar the oligosaccharide composition. And the further apart they are, the more dissimilar the oligosaccharide composition. So you see that is quite different and fills out a remarkable space here. These samples were collected really from all over the world, and we've seen that there's differences in different parts of the world. So what we are focusing our research on currently is to understand what actually drives this variation in human milk oligosaccharide composition between different women. What are the infant health and development implications? What do these oligosaccharides actually do for the infant? And then last but not least is, can we learn from what we discover in the infant maternal space and then have a deep mechanistic understanding how it works and develop therapeutics for really people of all ages? So this goes all the way from the preterm infant to people with cardiovascular disease. So today I'm only going to talk about infant health and development because that is really the topic of this symposium. What happens to oligosaccharides when the baby ingests oligosaccharides? They are remarkably stable and resistant to low pH and the pancreatic and brush border enzyme. But 1% gets absorbed and appears intact in the systemic and in the urine. And the rest gets either degraded by bacteria in the colon or gets excreted with feces and ends up in the diaper. Now, what do these oligosaccharides do? Here is a schematic that represents on the top a human milk oligosaccharide as an example, on the left a potentially good bacteria in green, and then on the right a potential pathogen in purple, and underneath is the epithelial cells in the intestine, and then immune cells underneath or in the in systemic. So we know, for example, that oligosaccharides serve as prebiotics. So they foster the growth of potentially beneficial bacteria in the intestine. But we think that oligosaccharides are more than just food for bugs. In fact, we have data to prove it. So we know that oligosaccharides are antimicrobials. Instead of fostering the growth of good bacteria, they can also stop the growth of bad bacteria. They're also known as anti-adhesives. And that means that many bacteria need to attach to the intestinal epithelial surface to be able to build a niche and hold on to the intestine and then eventually cause disease. And we know that oligosaccharides can block that interaction. We also have evidence that there's direct effects of oligosaccharides on the epithelial cells without bacteria being involved, but that response of the epithelial cells can now change microbial communities. And there's good evidence that human milk oligosaccharides affect immune cells, either in the intestine or on a systemic level. And even that can backfire into the intestine and change the microbial communities. So again, there's local effects in the gut, but since 1% gets absorbed, there's also systemic effects throughout the body and really in multiple different organs. And we truly think that we are just here at the tip of the iceberg, and there is so much to discover of what oligosaccharides do. Remember, there's 150, 200 different oligosaccharides, meaning 150 to 200 different opportunities to discover great things. How do we study human milk oligosaccharides? Well, we use spectrum from in silico, in vitro, and ex vivo models where we use either binding arrays or cell lines or organoids lately, move through animal models, and then use human cohort studies where we can test associations of different oligosaccharides with infant outcome, and eventually do clinical intervention studies in infants, but potentially also in adults. And all this is really connected in a network of multidisciplinary collaborators from around the world. Now, if we wanted to talk about HMO research and everything that's going on currently, it gets very blurry very quickly. So it's important to focus on just one topic. Otherwise, we can talk about on the preterm infant in the NICU and specifically focus on necrotizing enterocolitis. Necrotizing enterocolitis on NEC is a fairly common and very deadly disease of the preterm infant. About 5% of all very low birth weight infants develop NEC. And on the other hand, we know that preterm infants who receive human milk are at a 6 to 10-fold lower risk to develop NEC. Now, what is the connection here? Why is human milk so powerful in saving preterm lives? Is it potentially human milk oligosaccharides? So this is a graph that's developed for a review paper in Frontiers Pediatrics in 2018 and really encompasses the work of the last 12, 15 years. What we started in 2008 is we isolated human milk oligosaccharides from pooled human donor milk, tested that in an in vivo model in animals of necrotizing enterocolitis, and found that indeed human milk oligosaccharides have an effect in reducing pathology and increasing survival of the animals. We then went in and asked the question, which oligosaccharide is it? So we used a multidimensional chromatography approach to separate oligosaccharides first by charge, then by size, and then really go into epitopes and identified that one specific oligosaccharide called DSLMT is responsible for the protective effects in this animal model. Well, rats are not humans, so how do we now translate that to the human preterm infant? What we did here is we designed a mother-infant pair cohort in North America, so US and Canada, where we recruited specifically moms that had very preterm infants, and then collected milk samples that went into those infants, those preterm infants, almost every second day. We then analyzed the oligosaccharide composition in these samples and looked at, is there any associations between human milk oligosaccharides and the risk of developing necrotizing enterocolitis? And indeed, we found a match, meaning that the same oligosaccharide that we identified to be protective in the animal model, DSLNT, was at much and significantly lower concentrations in the breast milk that was given to infants that developed NEC. So it's protective in the animal model, but it's it is associated with risk in the human cohort as well. So we've done this in three different cohorts throughout the years. One in North America that I mentioned here, one in South Africa, and one recently in the UK. And in every case, DSLNT pops up as the significant oligosaccharide. So that helps us really inform clinical trials and also allows us to keep studying the mechanism in this animal model. So it's DSLNT, one specific oligosaccharide that is associated with lower NEC risk in human preterm infants, and the same oligosaccharides improves survival and pathology in an animal model. There's a great opportunity to develop new therapeutics, but it's also a great opportunity to screen mom's own milk or donor milk products for this specific oligosaccharide. If it is low in this specific mom, and remember, different moms make different oligosaccharides in different concentrations, if it's low in mom's own milk or in donor milk product batches, don't feed that milk to an infant that is at risk for NEC. Okay, so but how does this all work? This is data now from an animal model. This is data from human cohort studies, but we don't really know what the mechanism is. So what of this laundry list here on this slide is prebiotics and antiadhesives? Well, if it's prebiotic effects, and that's what many people think that oligosaccharides do mostly, then we always talk about a probiotic. So that's bacteria that we feed and that we want to have that utilize all oligosaccharides. In fact, there's a great hype around the bifidobacteria that metabolize all these oligosaccharides, and they're designed to utilize oligosaccharides, and that's how they benefit the infant. But if you think about it, if you have bacteria that utilize all human milk oligosaccharides, all these others can do, they can no longer do because now they're utilized, right? So if they're gone, they can't be doing all the other things. So maybe we want to have some probiotics that use some HMOs, but not others. And I'll show you some work that we've done there recently where we look specifically for probiotics and what kind of oligosaccharides they utilize. So here's the first example. Here is one strain of a probiotic, just call it probiotic A. And we see that this probiotic does not utilize any human milk oligosaccharides. So there is no advantage of having oligosaccharides here. Then we have a second probiotic. This is probiotic B, and this probiotic really utilizes almost all oligosaccharides. So you see there is a lot of oligosaccharides that go down in the chromatograms here, including DSLNT. So if we feed this probiotic, it gets rid of DSLNT. DSLNT is the protective oligosaccharide against NEC, so we would have a problem. And then there's other peaks that pop up, and we don't really know what they do. So yes, this probiotic utilizes many HMOs, but it also utilizes the potentially beneficial oligosaccharide against NEC. Now here is a third probiotic, probiotic C, and that only utilizes LNT. You see that is almost entirely gone, whereas you see that DSLNT is entirely intact. So the protective oligosaccharide is still there. So you see, trying to develop new pre and pro and symbiotics, you really have to see how these things fit together. And knowing which oligosaccharide is doing what in which context now allows you to develop probiotics that do similar things, and they can work in synergy. Okay, so it's all about developing targeted probiotics that utilize some human milk oligosaccharides and leave others intact. So this is just, well, now let's zoom out again and talk about all the other things, limitless opportunities. And for example, we work on anti-adhesive, antimicrobial effects, and so do others. Still a huge problem in the world, losing about 2,200 infants and children under the age of 5 to diarrheal disease. So there's great work that 2FL, for example, protects from some of these pathogens. And that's been shown in tissue culture and in association studies. LNT, does something similar for group B streptococcus. We've seen some work on Entamoeba histolytica from our own work. And then 3SL and 6SL have effects on rotavirus. So different oligosaccharides, again, target different pathogens. In the infant growth space, we've seen that 2FL is associated with growth in infancy and early childhood. Neo-LNT is involved in this as well, and the ratio between these two oligosaccharides seems to be important. And we've seen this over and over. There's a fourth study now that we're just working on right now from Australia, where we see the same oligosaccharides popping up to be associated with growth and development. And then there's data on 3SL and 6SL as well when it comes to malnutrition. More data on neurodevelopment and cognition. Again, 2FL standing out for memory and learning in animal models, but also we've seen this now in a cohort that we were involved in, that 2FL is associated with better cognitive development. And then 3SL and 6SL are oligosaccharides that are associated with memory and learning. So again, you see there's different outcomes, different developmental outcomes we're looking at, and different oligosaccharides that do different things. Structure determines function, and there is a lot of opportunities here. Last but not least, immune development, defocusyl lactose listed here related to cytokine release. 3SL, we've done a lot of work around 3SL in the chronic inflammation space. And there's a review that you can that I refer you to in Frontiers in Pediatrics that outlines really all the different oligosaccharides and what they do in the immune space. And then we notice that when it comes to immune development and immune modulation, it's very often a mix of different oligosaccharides that is important. So for example, for cow's milk allergy, it's not one oligosaccharide that needs to be there to protect. It's a mix of different oligosaccharides. Same is true for food sensitization. And recently we have a paper in Jackie here that looks at allergic disease. And again, it's the mixture of different oligosaccharides and not individual ones. So to summarize, human milk oligosaccharides, what are they and what they do? HMOs are the third most abundant solid component of human milk. And I think that is often underappreciated. HMOs are a group of complex sugars, glycans, and there are about 150 different ones. There's small oligosaccharides like 2FL, 3FL, 3SL, and 6SL. And then there's larger ones. There is neutral oligosaccharides that do not carry sialic acid. And then there is what we call acidic oligosaccharides because they are sialated, so carry a negative charge. And then there's some that are fucosylated and others are non-fucosylated. So it gets fairly complex pretty quickly, but maybe this list helps sort oligosaccharides in these different groups. HMO structure determines HMO function. I cannot say that often enough. Not all HMOs do the same thing. There is a reason why we have 150, 200 different ones, and different oligosaccharides do different things. They're not digested by the baby. There is local effects in the gut, but also there are systemic effects after absorption. HMOs are prebiotics, but so more than just food for bugs. They're anti-adhesives, antimicrobials, epithelial cell modulators, immune cell modulators, and probably a lot more. So they can affect outcomes like necrotizing enterocolitis was my prime example, but also infectious diseases and diarrhea, growth, neurodevelopment and cognition, and certainly immune system development and response. And quite frankly, we still think that we're at the very tip of this, and there is a lot more to discover. So what we see that sometimes you need specific human milk oligosaccharides to be effective. So one structure is important, like DSLNT, and you cannot substitute that with another oligosaccharide. So highly structure specific and likely receptor mediated. And then sometimes, like in the allergy space, you need mixtures of different oligosaccharides to be effective. And here it's the relative abundance, the ratios of different oligosaccharides that really matters. And the mechanisms are likely indirect to either shaping microbial communities or shaping immune system mechanisms as a complex system and not just as one receptor, one oligosaccharide interaction. So in any case, we need a combination of preclinical, cohort, and clinical trials to fully assess what these oligosaccharides do and what the functions are.